Good afternoon, everyone, and welcome to BioAlberta's mentorship series. I'm Rob Stoddard. I'm the president and CEO of BioAlberta, and I want to thank you for joining us today. BioAlberta is a member-driven and funded voice for Alberta's life sciences sector. Our mission is to grow a vibrant life sciences sector to diversify the economy by leading Alberta's recovery. We support and connect researchers, students, SMEs, industry leaders, and funders to advocate for our success. Events like today's webinar are part of a mentorship series which offers access and insight from industry leaders who share their expertise and experience to help our sector grow. Today's webinar is part of a series of 4 sessions by the Metabolomics Innovation Center at the University of Alberta, focusing on metabolomics and the life sciences. Our workshop topic today will focus on precision medicine and how metabolomics, supports Alberta's leading edge companies. Before we get too far into the session, just some housekeeping items. If your microphone isn't muted, please mute it just to keep some of the background noise down. We do have some questions already submitted, but if you would like to ask a question, we'd ask that you put it in the chat, and then we will ask it at the appropriate time. And now on with our session. I expect many of you already know Dr. David Wishart, which is why you're here today as part of our mentorship series. Dr. Wishart, who's received his PhD from Yale in 1991, is a distinguished university professor in the departments of biological sciences and computing sciences at the University of Alberta. He's also co-director of the Metabolomics Innovation Center, Canada's national metabolomics laboratory. He is consistently one of the world's most highly cited scientists, and for good reason. He has published more than 430 scientific papers covering many areas of protein science, including structural biology, protein metabolism, and computational biochemistry, and has been cited more than 78,000 times. Dr. Wishart, I want to thank you for leading today's presentation and discussion. Before I turn the screen and microphone over to you, I want to introduce your 3 colleagues who will join us, starting with Ian. Guys, if you want to unmute yourselves and introduce yourselves quickly, we'll go right back to Dr. Wishart.
Sorry, hunting for the Zoom button here. It's my first time ever on a Zoom conference. So. My name's Ian Lewis. I'm an associate professor here at the University of Calgary, and we specialize in using mass spectrometry-based metabolomics approaches for developing new precision infection management strategies. And our program recently has launched a couple of new technologies for improving the management of bloodstream and urinary tract infections. And yeah, we're fascinated in the interface of metabolism and infectious diseases.
Julian.
Hi, thank you, Rob, for the introduction and for this opportunity to participate in today's session. My name is Julian Lum. I'm a senior scientist at BC Cancer in Victoria, associate professor at the University of Victoria. My research program is very patient-centric and focused. We use metabolomics as a tool to discover new hits and targets with the idea and the expectation that we can use those to design customized cell-based immunotherapies for cancer. For example, engineering strategies that would enhance the cancer-fighting properties of chimeric antigen or CAR T cells. Secondarily, we're quite interested in using metabolomics as a tool to help us identify individual metabolomic profiles that would help us to design interventions, dietary or nutritional ones, that would help their immune responses to cancer. So thank you, and I look forward to exciting discussions today.
All yours, Rob.
Thank you. Hi, I'm Rob Fraser. I'm president and CEO and co-founder of Molecular U, which we co-founded with Dr. Wishart. We have a footprint in Alberta, primarily working closely with Dr. Wishart, and one in Vancouver and in Toronto as well. Our focus is really about bringing multi-omic technologies to practice. We are one of the few companies in the world that are employing multiple omics to help us really understand where an individual's health is and what can be done about it. We find the metabolomics key in this multi-omic description of your health. It's very important that we understand the mechanisms that are going on to really understand how we can prevent or help comorbidities from emerging in complex disease situations. We're applying our technology primarily in the preventive scheme, working with groups in corporate health, but we're also finding that there's great applications in patient support programs. And so I'm very, very pleased and honored to be here today and with such an esteemed panel. And thank you for the invitation. And I hope you enjoy the discussion.
Thanks, Rob. David, the screen and microphone are all yours if you want to unmute, and we're in your hands.
Super. Thanks a lot, Rob. And welcome, everyone. Thank you for joining us today. I'm going to my screen up, go to full view here, and hopefully everyone can hear and see the slides. So as advertised, we'll be talking about metabolomics and precision medicine, and try and give you some both background and some examples to show how metabolomics can make a difference. I'll be speaking for about 30 minutes, and then the last 25 minutes or so will be a panel discussion, and hopefully everyone will have questions, and we can sort of field your thoughts and suggestions and questions after this is completed. So as I said, I'll just introduce things. Many of you probably have heard about metabolomics. Some of you maybe already attended the previous session, but this is just sort of a quick update in case those of you are new or coming to this series for the first time. So the metabolome is what we call the full collection of the small molecules found in either biological or environmental samples. So small molecules can be things like amino acids and nucleic acids, organic acids, sugars. They're the things, the building blocks of the cells. They're the fuel that keeps living systems alive. But it can also include other things. So we call the endogenous compounds part of the metabolome, but they're also exogenous things, things that you may eat if you've just had lunch. You're bringing in a variety of phytochemicals or exotic plant compounds, the natural products and food additives. If you drove into work today, you probably inhaled some pollutants. If you may have been taking some drugs to treat a condition, or there may be some cosmetics. All of those compounds actually end up eventually in your bloodstream or in urine or in other excreta. And all of those are part of the metabolome. Some of them are good, some of them are bad, but they are the chemicals that we find in not only ourselves, but we can find them in fish and flowers and other plants, crops and water and soil. And so this is part of the matrix that we look at when we do metabolomics. Another way of looking at metabolomics is through this pyramid that I call the pyramid of life. You know, the genes represent the genome. We use genomics to study them. The base of the pyramid is occupied by the genome. Genes code for proteins. Proteins, a complete collection of proteins is called the proteome. We use proteomics to study the proteome. And then proteins either manipulate, transform, enzymatically catalyze the production of metabolites. And the whole collection of those is, of course, the metabolome. And the study is metabolomics. As you climb up this pyramid, there's different levels of influence, both with physiology and environment. So the genome is remarkably stable, and it's supposed to be stable, literally for hundreds of thousands, if not even millions of years. But stability of the metabolome or proteome is somewhat less. We also know that the genome in every cell in your body is identical, but the metabolome in different cells and different tissues and different organs is actually quite different. The metabolome of the brain is different than the metabolome of the heart. and different than the metabolome of the liver, different from the metabolome of urine versus blood. So what the metabolome is it lies at the interface between the genome and the environment. And the fact that it lies at that interface means it's really useful for informing or getting information about both the genome and the environment. And that's the two of them are what define the phenotype or the phenome. So metabolomics has picked up a lot of interest over the years because of the fact that it lies at that interface. Now, as mentioned by Rob and others, I have been working with the Metabolomics Innovation Center, or what we call TMIC. This is a facility that started almost, 10 years ago. Originally, there was 3 nodes in Alberta and Victoria, and then since then it's grown to 8 nodes, including Ian Lewis, who's on the panel today. And there are now 35 staff, almost $40 million in equipment, and that's being added to almost on a monthly basis. TMIC is a facility, a set of nodes and labs designed to support both metabolomic service, but also technology development. It's funded by Genome Canada and as well as by the CFI. This just illustrates how it's spread out across Canada. So even though we call it a laboratory, it's a virtual lab, but it's connected through many nodes. There are 5 nodes in Alberta. There's a node in Calgary with Ian Lewis. There's a node in Victoria with David Goodlett, a node in McMaster University with Phil Britz-McKibben, and a node at McGill University with Christoph Borchers. And each of the nodes specializes in certain areas of metabolomics, but we're all connected through lab information management systems, common SOPs and protocols to ensure that the processes that we follow are consistent and accurate and reproducible. So that's a bit of a background on metabolomics. Today's topic obviously is not just about metabolomics, it's about precision medicine. And many of you have also heard about precision medicine, and this is just a simple definition. But what it really focuses on is the idea of both treatment and prevention that looks at the variability of individuals at what the genome, the proteome, the metabolome level, but also their own environment and lifestyle. Each person is unique, and we're unique not only from our DNA, but unique from the perspective of how we live our lives, where we live, what we eat and drink, and those are all reflected in the genome, proteome, and metabolome. With precision medicine, there's also kind of a shift away from sort of reactive medicine to proactive medicine, with the idea of disease prevention, early diagnosis, but also facilitating more rapid drug development and looking towards a personalized treatment and wellness and health through proactive health monitoring. So there's a lot sort of in this slide in terms of the concept of precision medicine, but as I say, it's changing from reactive medicine to proactive medicine. Now, precision medicine has been around for a while, and people are probably more familiar with precision medicine as it's applied to cancer, particularly with things like the HER2 test or the BRCA1, BRCA2 tests. These were designed in some cases like the HER2 to decide on status, or the BRCA1 with the idea of prevention. And these are, genetic tests that could be performed on blood or tissue samples and allow physicians and patients or would-be patients to make decisions. And some of these were guiding the treatment, or others were for preventing disease. So genomics was the first, I think, to really apply precision medicine and to have real useful examples for that. Now, metabolomics is maybe a little bit of a latecomer, but I think it's important to distinguish what metabolomics can do. Whereas the genome can tell you what might happen to you, the metabolome tells you what is happening. It's giving you a real-time readout. So we can take your genome and look for a mutation in the BRCA1 gene, and we might say, 30 to 40 years from now, you might develop breast cancer. But with a metabolome, we can actually do a blood sample and say, you have breast cancer. And so this is the difference between what might be and what is. The other thing about metabolomics and metabolites in general is that these things are actually fairly easy to change. Metabolites you can adjust and modify with your diet or with certain drugs or with supplements. It's really hard to rewire your genes. You're born with them. There's gene therapy, but that's not really designed to change every gene in your body. The other thing that's happening is that metabolomics is getting faster, it's getting cheaper, it's highly quantitative, getting much more comprehensive, and the data is actually becoming easier and easier to interpret. So this makes metabolomics nicely suited for precision medicine. And there's also lots and lots of useful metabolite biomarkers that are known. These are some numbers that we had collected from clinical assays and studies reported in Western Canada. So it's not universal, but it's probably a good sampling. So if you look at the number of approved tests that are using clinical chemistry or that measure metabolites using various clinical analyzers, there's almost 350 compounds. In terms of the number of approved tests that are used or using genomic assays, there's about 130. Number of approved protein tests that use ELISA methods is 108. Technically, there aren't any approved transcriptomic tests in Western Canada, but globally there are 5. And currently there are no officially approved proteomics tests that use mass spec. It doesn't mean that proteins aren't useful. As I said, the ELISA tests measure about 108 biomarkers, but just using mass spec as a tool for proteomic or clinical biomarkers just has not picked up. So from this list, you can see that metabolomics or clinical measurements of chemistry or compounds is actually the most widely used method for detecting biomarkers of disease. Now, one example where metabolomics and precision medicine have actually been happening for a long, long time is in newborn screening. If you've ever had a child or if you're under the age of 30, you've probably gone through newborn screening. And this is a heel prick where blood is collected shortly within a few hours after a baby's born. And a blood spot is collected. It's then sent to a mass spectrometer and it screens for amino acids, organic acids, and acyl carnitines. About 25 to 30 different metabolic disorders are checked. Newborn screening is widespread. Every province in Canada offers it, every state in the US. And anyone who's under the age of 30 probably has had a metabolomic test. You just don't remember it because you were a baby. And most parents don't know it because it's done as a routine screen for all newborns shortly after the baby's been born. But this is one of the most successful examples of an omics test making it into the mainstream in terms of newborn screening. And the reason why it's, I think, such a useful example for precision medicine is it shows how metabolomics can give you precise and personalized diagnoses as well as prognoses. And in some cases for up to 50 different inborn errors of metabolism. So it can say, yes, you have PKU, or yes, you have MSUD, or no, you don't have any of these. And it can say whether it's going to be severe form or a mild form. Newborn screening catches the conditions very early, literally hours after you're born, so that they can intervene with precise diets or lifestyle changes or medical foods that are given to the infant to ensure that they can develop normally. And what's more, even after the diagnosis has been made, metabolomics continues to play a role throughout that person's life, helping to monitor or help to mitigate their condition. It's done first by the parents, and then as the individual grows as an adult, and they use these metabolomic tests to make sure that things are well under control. So newborn screening has already been applied to more than 300 million infants. And it's saved the lives or improved the lives for an estimated 1 million people. So this is a huge impact that metabolomics has had, but it's one that almost no one knows about. There are examples of these precise medical foods that have been created. Many of them were developed actually in Canada initially, but they've been spread around the world. So they have obscure names, BCAD or Phenyl aid, but they are essentially modified foods that allow you to treat people with conditions like phenylketonuria or maple syrup urine disease or citrullemia. And the diets are, easy to prepare. They're a bit of work, but in some cases you just supplement someone with biotin, or you supplement with carnitine, or you make sure that the diet is low in phenylalanine. And these things, I say, have profound effects. People put on the diets can develop normally and lead normal lives all through the development of these metabolomic assays. So inborn errors of metabolism, I think, are a good example of the application of precision medicine and metabolomics. There's another example, and I'll give, I think, about 4 or 5 examples in this meeting. And one is the application of precision medicine towards accelerated drug development. So we've been working with a company called OMX, or Omics, and they're responsible for database drug bank.
Like Alex played FIFA after school, and I didn't want to do that, so guess what I did?
Sandy, do you mind turning your microphone to mute, please? Anyways, what you can do with metabolomics is not only identify drug targets, but you can also use metabolites as drugs or drug leads themselves. And this is an effort, as I said, that started with TMIC a number of years ago, and then it's led to a spin-off company, which now employs about 45 people in Edmonton. Traditionally, the way that drug discovery goes is that we usually start with a genetic or genome-wide assessment. We identify genes of interest. And from the genes of interest, then we'll identify a protein target. From the protein targets, those will be cloned, and then we'll start using chemical libraries to see if we can enhance or inhibit that protein target. And that's the drug discovery process. And I've given the odds of success in terms of finding a gene, cloning a protein, then finding a potential lead compound. And that's the phase 1 or discovery phase. But from there, there's a lot of other work that involves testing, validating later stage clinical trials. And those can take a long time, but they also have a very, very low chance of success. And even among approved drugs, approximately 1/2 will sort of fail the phase 4 trial, which means that either they're not as good as everyone had hoped, or that they lead to adverse drug responses, and so they have to be taken off the market, or the sales never meet what people expected to meet, so they're pulled by the drug company. If you look at all the odds, the chance of actually finding a drug from a GWAS, a genome-wide study, is about 0.001%. On average, it takes about 20 years from discovery to completion, and costs between $1 and $1.5 billion to bring the idea to fruition. So it's one of the reasons why the drug pipelines are drying up. On the other hand, a number of approaches have been taken towards trying to use metabolomics rather than, say, genomics to help with drug discovery. Metabolome-wide association studies can be done very inexpensively, and they can associate changes with metabolites and disease. The analysis of metabolomic data is very quick. It allows you to immediately go to analysis of pathways. And because many of these pathways are well known, you can use data resources like DrugBank and other tools to identify either inhibitory metabolites or inhibitory metabolite-like compounds to just potentially use as drug leads. Of course, there's testing and monitoring, and in many cases, sometimes the drugs can be used simply as supplements. They don't have to be invented drugs. They can be, as I say, a change in diet, a modification in supplements. And then with metabolomics, you can also continue to monitor. So overall, metabolite-based drug discovery appears to be both cheaper and faster, and a number of companies are starting to do this. One example was actually the development of a drug for glioblastoma. Metabolomics was used to discover a particular compound, 2-hydroxyglutarate, which is elevated in gliomas or glioblastomas. So this is a discovery catalyzed by metabolomics. It was published in 2009. It took a number of years to sort of figure out how this mutation led to this hydroxybutyrate and what the proteins were involved. Essentially, it was isocitrate dehydrogenase. But through that metabolomic discovery and using sort of this metabolite-based or metabolomics-based drug discovery, a new drug was developed by 2017. So 2009 from discovery, they weren't even thinking about drug development, to 2017 to the creation of a drug. That used a lot of the concepts that I just illustrated for metabolomics-based drug discovery. And there have been others that have essentially emerged from the same observation about isocitrate dehydrogenase inhibitors. So this is a really, I think, interesting story about how metabolomics has enabled precision drug development for a very specific form of glioblastoma caused by a fairly specific type of mutation with isocitrate dehydrogenase. Another example also related to cancer comes from work from Metabolomics Technologies Inc., or MTI, and from one of the founders of MTI, Richard Fedorik. A number of years ago, he was a gastroenterologist, and he worked on the field of colon cancer and colon polyps. Colon cancer in North America is a very common disease. It's one of the leading causes of death. But if you get colon cancer at stage 0 or 1, the cure rates are about 100%. So early prevention makes a big difference in colon cancer. Now, most people know how you screen for it. There's colonoscopies. There's also Cologuard or fecal tests, fecal immune tests. But those have very poor compliance rates, around 5%. So the question that Richard asked was, is there a better, less invasive way of detecting for colon cancer? Colonoscopies are very invasive, and fecal tests have a very low compliance rate. So Richard decided to look for urine markers for colon cancer, colon polyps. And so through studying both urine, looking initially through NMR, and then later through mass spectrometry, was able to detect markers for polyps, and 3 metabolites, carnitine, succinate, and ascorbate, showed up as being strong markers. And we worked with MTI to help develop a mass spec-based test that would be used. And this is now called PolypDX. And it's now used in labs as a diagnostic test, or a lab-developed test, LDT, for several diagnostic labs in the US, ADL and Lab Express. So this has been going on since 2018. TMIC and MTI are continuing to work in partnership. As MTI is based in Edmonton, developing new markers for colon cancer. So that's different than polyps, and there are different markers for colon cancer. And then trying to extend the work to look for additional markers that will improve the sensitivity and specificity of the polyp test itself. I'm talking a lot about cancer, but it just happens to be where I think a lot of the efforts in TMIC have been focused for the last couple of years for precision medicine. And this is, again, a collaboration that grew out of a company based in Vancouver called Biomark. And they are interested in lung cancer. Again, lung cancer is probably the worst cancer to get. So it has, it's the leading cause of cancer deaths in Europe and North America. Most lung cancers got it at stage 3 or 4, and typically survival rates are about 10%. So it's a pretty grim cancer, but they know that if they could catch lung cancer much earlier at stage 1, lung cancer is fairly curable. So this is another example of this idea of prevention or proactive medicine. If you could screen for lung cancer, we could save a lot of people's lives. CT, or computed tomography, X-ray scanning, is one of the new approaches people are trying to be able to identify small tumors in the lungs. You can see an example of a CT scanner and a small tumor identified in someone's lung. Now, CT scanning, just like colonoscopy, is expensive. It also exposes people to radiation, and it produces lots of false positives. And so again, the question is, can you come up with a better, less expensive way? And so we worked, as I said, with Biomark to look for a blood marker or set of blood markers that would allow you to detect lung cancer at very early stages, stage 1 and stage 2. Again, using mass spec, we were able to identify a number of markers, 5 metabolites, but also some clinical history. So if someone is a smoker, that's clearly someone who's at a higher risk of lung cancer. And we looked at cases, controls, we validated in several different ways. And the area under the curve, which is a way that we measure the quality of a biomarker, is about 93%. When we compare this metabolite test for detecting stage 1 and stage 2 cancer against the low dose computed tomography, we're about 10% better. So AUC of 0.93 with metabolomics, AUC of 0.83 for computed tomography. So the graph is shown on the left, and that's the kind of curve that you want to see when you are doing marker development. And so this is now being followed on with a larger cohort, and we'll be looking with patients who have other lung diseases like tuberculosis and pneumonia or COPD. So this is a way of rigorously testing to see if the marker is not just exclusive to lung inflammation. We will, if we validate this successfully, and we've been recently funded to do so, then obviously we'd like to publish the findings. After the validation step, there's further test refinement, LDT testing. You always have to do things like economic modeling, lobbying, and then eventually you have to go for Health Canada or FDA approval. So it's still a long ways away, probably another 5 or 6 years, but this is, I think, a promising example of using metabolomics to do proactive medicine and precise diagnoses to help in prevention and ultimately saving lives. Another area that's emerged, and you heard discussions with Rob Fraser talking about molecular U. This is the idea of wellness testing. And a number of companies have emerged over the last decade that are using genomics, proteomics, metabolomics, phenomics, microbiomics to assess a person's health status. So there's companies like Viome, Human Longevity Incorporation, or Whole Biome, and also Molecular U, which is the Canadian presence in this field. So Molecular U, as Rob said, was started in 2014. It has facilities in Vancouver, Edmonton, Montreal. And the focus is on both personalized health and precision medicine. It uses a lot of multi-omics measurements. It integrates things with AI or machine learning, big data analytics to ultimately provide some guidance. The tests that Molecular U does for metabolomics are done through TMIC facilities. And this allows them to get the quantitative data that they need to do their analytics. This is sort of a picture of the type of analysis that's done, where whether it's amino acids or acyl carnitines, they're compared to reference values that have been collected and assembled by Molecular U. And then things are identified as being above normal, normal, below normal. Things that are flagged in red say there's a problem. Things that are green say they're okay. And the combination of amino acids and acyl carnitines is often a very good indicator of whether someone is pre-diabetic. And this person actually is. You can see elevated levels of glucose, but we can also see telltale markers of the branched chain amino acids, isoleucine, leucine, phenylalanine, and then also elevated levels of short chain acyl carnitines, which also indicate sort of a pre-diabetic status. So this again is something that you can identify an individual before they develop the disease and then potentially intervene, but also monitor and ensure that they adjust their metabolite levels to bring them to the healthy levels that are needed. So Molecular U has a pipeline where, as I say, they not only measure metabolites, but they measure proteins and toxins and the microbiome and the phenome. And that's all integrated. through their health intelligence platform called MyHigh. And then it's not just simply a readout, but then there's action processes that are developed and guided through that, suggesting changes in exercise, nutrition, or supplements, or even medications that can be used to improve an individual's health and then prevent disease or enhance their well-being. So as part of this effort, working with Molecular U, TMIC has been adding ISO and CLIA certifications to comply with those sorts of industry needs. It's been developing custom exposome tests and metabolomics kits for MyCo's needs. And then it's also been participating in a variety of grants, partnerships through MyTax and NSERC, the Supercluster Initiative and Western Economic Diversification, both to train people through postdoctoral fellowships, to perform R&D research, looking at mild cognitive impairment, autism, cancer, and COVID. So this has been a nice partnership working with people in Alberta and with BC, and also extending to other parts across Canada. So this talk, as I said, is primarily focused on biomedical applications with precision medicine, but there are many areas where metabolomics can impact in newborn screening, health monitoring, drug discovery, chemical imaging, diagnostics, toxicity profiling, immunometabolomics. The list goes on. I obviously don't have time to talk about all of these applications, but you'll just have to trust me that these are widespread. And in fact, many of these are areas where TMIC is actively involved with researchers or with companies moving this forward. So there's lots of applications in biomedicine. In terms of what TMIC can do, I mean, part of it's giving you some of these examples, but we've worked with many groups, many companies, many organizations to help advise or offer advice for precision medicine or personalized wellness work. The ISO certification, CLIA certification, the services that are now offered, as well as both software and databases. A lot of expertise and a history of doing biomarker translation, moving from discovery to the clinic. And then I think to facilitate a lot of this, we've worked with a number of groups to provide them with small-scale pilot studies to help launch some of their ideas. Those pilot projects have often led to large-scale studies that involve support from multiple organizations. So I think there's a lot that TMIC, or the Metabolomics Innovation Center, can do in trying to facilitate business development in the area of precision medicine. If you want to know more about TMIC or about the things that we can do in the area of precision health and wellness, I'd invite you to contact Jen Reed, who is also online today. She's wearing two hats. She's not only the operations manager for TMIC, but she's also the business manager. And so the website is there, and Jen Reed's email is also there, as is my email at the beginning of the slides. As I say, this is not done by one person. There's many people, many components, and these are the node leaders at TMIC: Liang Li, Christoph Borchers, Phil Ritz-McKibben, Michael Overton, James Haranack, Dave Goodlett, and then Ian Lewis, who's here today on the panel. And then we've been supported by not only Genome Canada, but many others. So I think I am going to stop that and let us go back to our main group and I guess invite any questions. I think, Rob, is your audio on?
Thanks for that, Dr. Wishart. Before we get started with the panel, while they find their unmute button, since I couldn't, let's get the audience participation marks out of the way. If you could find your raise hand button, I'd be interested to know how many of you have sent in a DNA test kit, like a 23andMe or an Ancestry type question. Just a quick show of hands in the participant box would be helpful. Okay, it's either a really shy group.
They don't believe in genetics.
All right.
We weren't going to mock them, were we? If you did it.
No.
Offered a prize. Maybe we would have. Would have got better participation. So, all right, 0 audience participation marks for everybody today. So we'll start with a couple questions, and then we've got a couple coming in on the chat. Start off, and maybe this is to Rob and Ian, but anybody on the panel. What are the biggest challenges companies in Canada face when bringing new precision health technologies to market?
I'd be happy to begin. So there are multiple challenges to starting any company in Canada, but I don't think there's anything unique about the precision health in that. One of the great, though, when we're talking about preventive care, is who should pay. And most of the healthcare in Canada is covered by our provincial healthcare systems, and so it's a challenge to get people to be active about their health in a preventive capacity, and most citizens in Canada feel that the Canadian healthcare system will look after them when they're unwell, and that's true for the most part. And most people don't get activated about their health until they're sick. So I think those are the 2 challenges in a precision health that are unique to precision health. There's lots of other challenges, though, but I don't think we have time for all of those.
Okay, a question from the audience. In the near future of precision medicine, do you think that using metabolomics alone, exclusive from analyzing other areas of the pyramid like genomics and proteomics, will be effective by itself?
Maybe I could answer, and maybe Ian Lewis could also bring in some comments, others too. But I think my own view is that it helps to integrate things, that you shouldn't really be an island in the world of omics. There are some real strengths where metabolomics gives you insights that genomics or proteomics can't for certain disorders. But then there are other disorders where genomics gives you insights metabolomics can't. So I think, we want to use all the tools in our tool chest when it comes to precision medicine. But we also want to use the right tool for the right condition, and don't use a screwdriver as a hammer sort of thing. I don't know, Rob, Ian, Julian, you guys might have some other comments.
Yeah, I think that's absolutely correct. We find that in certain conditions, taking a multi-omic approach is absolutely required insights in the individual's health. But there are certainly instances where multi-omics, metabolomics is sufficient. And so we started to explore a little bit more the urine, where proteomics and genomics really isn't very, can't even be measured, to be honest. And so there's a lot of value in the urine metabolomics that I think is actually quite exciting and emerging as a great diagnostic tool.
Okay, thanks Uriel Perez for that question. Next question from, and I hope I pronounced the names even close, from Ingrid Casido. Do you work with mental health metabolomics?
So we do. We've worked in areas of autism. We've worked in looking at anxiety and depression. We've looked in Alzheimer's and Parkinson's within our own studies. Rob in Molecular U has also done some stuff, but maybe I should just ask, because Rob and I are answering, so maybe Julian and Ian could also comment on some of the areas if they've worked in mental health and metabolomics.
I mean, we have, there's been a long history of connections between mental health and metabolomics, as Dr. Wishart pointed out. There's been a lot of interested in that transect, and now increasingly in the gut microbiome, metabolomics, mental health kind of access too. So yeah, I think a number of us have worked in that area. There have been some interesting developments. I think it's still an area that's being actively explored. It hasn't been hammered down yet, but there's certainly a lot of history there, and some really tangible things that we've been working on include things at the interface between pharmacokinetics and gut microbiome interactions, and how those interactions can modulate the effective pharmacokinetics of some of the drugs that are used for mental health reasons. So that's a specific example.
Okay, thanks for those answers. From Marissa Jones, what are the biggest roadblocks to bringing a tool from research purposes to a diagnostic tool?
I guess this is maybe a good one for me. So we work at this axis. That's sort of our specialty, is trying to cross that bridge between research and implementation. And so it's a big gulf. There's a big gulf in mindset, and there's a big gulf in terms of what constitutes completion. So just discovering things is fine in research. A product, you kind of have to take things to many orders of magnitude beyond when it comes to proof and supply chain and working with regulatory frameworks. And so it's a very, very long and difficult process that translates things from a well-known discovery into a usable tool. As a relative newcomer to the field, it never ceases to amaze me how much work goes into real-world products. You should be very thankful anytime you pick up a glucometer or a pregnancy test strip or any one of those devices. You have no idea how many hundreds of thousands of hours went into those devices. But yeah, so it's a long road, but there's a direct connection between the things that we do in metabolomics, genomics, and proteomics and real world tests. Like the people who are working in academics, provide the discoveries that become tomorrow's diagnostic tests. And but there's just a huge amount of effort that's involved in the translation.
Thanks, Ian. Anybody else want to jump in?
I think I would just echo with what Ian has said. It's an enormous amount beyond the discovery phase to do the translation, and that you often bring in a lot of engineering, a lot of quality assessment and quality management, and these are things that are not generally taught at universities, it's an important thing for industry to understand, and it's something that universities need to probably push in that regard to help train those, because it's those people that are critical for translation and for the success of industry.
And the only thing I would add is that, at the end of the road, the reward for diagnostics is not huge. So there's all these hours of work that put into it. But the profit margins are actually quite small. And so this is one of the reasons we focused more on the wellness area and stay more in the gray space of care, because we can start to work now and deliver information without being diagnostic.
All right, thanks to the panel. From Hussein, can we use metabolites for renal hypertension?
Yes, you can measure a variety of metabolites, not only for hypertension, but aspects of renal failure or other renal diseases. You can measure both blood and urine for metabolites that are associated with renal conditions. And they have been used not only to identify conditions like renal hypertension or uremia or renal failure, but also to monitor things like kidney transplants as well. So the kidneys and urine can provide a lot of information about what's going on. And obviously there's a direct connection between kidneys and urine.
All right, question from Paul. Please, I would like to know the step in drug discovery that involves use of metabolomics. Could it be an indicator for the choice of active pharmaceutical ingredient or clinical testing?
So the slide that I had was sort of illustrating that you can use metabolomics in many steps, both for identifying a drug target, identifying a drug lead, assessing drug performance, assessing drug safety or toxicity. So you can, maybe it wasn't clear enough or I didn't spend enough time, but technically metabolomics can be used in every step of the drug development, drug discovery process. And some of the larger pharmaceutical companies are actually doing so. They have it integral to every part from preclinical to phase 4. And that's, I think, one of the real appealing things about metabolomics because it can be used throughout the drug discovery development process.
Yeah, we're finding some interesting applications also in the care as so even post-market work. So often when a pharmaceutical company will launch a new drug, they want to enter it into the market, and so they try to get it into patient populations in these organizations called patient support programs. And through that, there's a lot of interest in further monitoring how the medication responds in a real world setting. And so we're finding some very interesting applications sort of post drug discovery and maybe even more in that phase 4. And this will help too in taking those learnings and improving on the profile of the drug to either improve its safety or its efficacy, but also understanding the differences of how individuals respond to certain treatments.
Another question, going back to the cancer detection examples in the presentation, are these alternative or novel disease detection metabolomic technologies widely accepted by physicians and sciences compared to traditional technologies, or do you envision more marketing is required? Marketing is in quotes.
I think there's a bit of both, probably. There's a fair bit of research that still needs to be done. One of the challenges you have that we found with the sensitivity of the metabolomics approach is that cancer is a funny thing. So everybody generates cancer cells throughout their lives, and whether or not it's a dangerous event, the thing it tends to take many genetic changes for that cancer to really take hold and to become a danger to your life. But we all experience cancerous moments. And so the challenge is that sometimes the metabolomics assays can be so sensitive. So we have to come up with a whole other protocol. So it's not just a matter of marketing and detection. It's working closely with the physicians and understanding what these technologies can bring to bear. Because if I see cancer in my profile, I don't want to panic. I just want to know that this happens and we're going to watch this. Most cancers take a long time to develop too. Pancreatic cancer is expected to take about 10 to 15 years before it fully flourishes and becomes a dangerous thing. And yet, it's seen as one of the most deadliest cancers because we don't detect it until very late. So there's real advantages to kind of this monitoring and so on, but you don't want to put people into a panic unnecessarily. That's kind of the challenge that we've found with introducing them. We are pretty excited about a project we're about to launch in cancer recurrence. So this will be working with people that have had cancer and so more likely to have a cancer event and then use the technology there to determine if we can detect it earlier so that we can have an earlier intervention in a recurring cancer.
Yeah, maybe I could add a couple of words on this. Seeing I do, at least my basic science research lab is in the cancer space. So I think your question is, physician or scientist uptake regards to cancer. And, I think we have known for many decades now through original work by a German scientist, Otto Warburg, that cancer has a very different metabolic profile than normal tissues. And we've developed actually a test, an imaging test, which is called 18-fluorine deoxyglucose. It's a PET scan, so it's an imaging test that at a very gross level will allow physicians to see which cells, or the tumor cells, taking up glucose as a fuel. And so I think that physicians and scientists alike do understand the value of these metabolic perturbations that happen in cancer. I guess the real question is, how do you develop an assay based on metabolomics to be able to detect what I would call the tipping point, right? So you're healthy, but at some point in your lifetime, that healthy profile begins to tip towards the cancer. So I think it's perhaps not necessarily a question of more marketing or the need to convince clinicians and practitioners that metabolomics technologies are important. I think the onus is on us to develop the technologies so that the detection is sensitive and specific enough. And I think some of the examples that Dr. Wishart gave, for example, the IDH mutation one is a really good example of how the metabolomics assay was able to achieve that sensitivity and specificity for that specific kind of cancer.
Thanks for that, Julian. So we have a request to please arrange an online lecture on Metabo-Analyst and how to analyze data on it. So David, you and I have homework, I guess. We're gonna have to figure out how to provide that to the audience as well.
I guess so. Actually, I could comment that we do have a workshop series that's, I think it's scheduled in June to do the Metabo-Analyst. And then there is also a video that's available through TMIC to do, that describes some of the analytical capabilities. I guess it's an animated audio PowerPoint presentation that we've posted online on the TMIC website. So there's several routes, and the Canadian Bioinformatics Workshop also has videos explaining how to use Metabo-Analyst, and they're about 3 hours of explanations. So those are some of the resources that TMIC helps produce to help people.
Perfect. There you go, Hussein. You can go to the TMIC website, or I think I heard earlier today that TMIC is launched on LinkedIn as well. So you should be able to find the resources you need. One final question. We're just about out of time, but if you do have the opportunity, or do have one final question, please put it in the chat while we ask this one from Marissa. Thank you for the great talk. How do you account for metabolic differences amongst populations based on race, environment, etc., when determining a biomarker, for instance, a biomarker of prediabetes?
So I think Ian and Rob have probably had experience in this as well. But typically, when you're doing biomarker studies, you try and do studies of diverse populations. You'll do tests in sometimes different countries. You will try and collect reference values for people of all ages and genders and race. So the validation process of once a biomarker has been initially discovered is difficult, and it is exactly that, where you're looking to see how robust the marker is relative to different populations, different age groups, even different diets. And this is where about half to 70% of biomarkers fail. Is that when you try and do the validation on the larger populations, you find that marker just didn't pan out. And so that's why validation is so important, to move a marker from discovery to the stage where it's a lab developed test. In the case of the MTI markers, those were tested in Asia and North America in large groups. So those have been well validated. But for prediabetes, there seems to be a general identification with these have been detected in populations in Europe, North America, and Asia quite consistently. So these branched chain amino acid ones are considered universal and very robust.
Yeah, I don't have too much to add other than we have looked at very diverse populations, and what we found is there's individual differences that are sort of across the board, that there's a lot of uniqueness in everybody we look at. And so we really do think of it more as a personal approach, and rather than a divided up into different cultures or races. So we take a much more personal approach to it. I think it's probably the best way we look at it. But as David mentioned, many of the things that we look at have been validated across very diverse populations.
All right. So that's it for the questions from the audience. Thanks everybody for your time and attendance today and the questions you've asked. David or Julian or Ian or Rob, are there any final comments you'd like to make, or final pitch to the participants?
We just want to pass on our thanks to everyone who's attended today. We know it's a busy day and you have other things to do, so we really appreciate the fact that people came today, and I certainly want to pass on my thanks to all the panelists and the organizers for helping putting this together.
Yes, thank you, everyone. Really great session, and I think the Metabolomics has a very bright future in how we improve our health. So thank you for joining us.
Yep, same thing. And just remember, the next time you sit down and have your favorite meal, you think about your metabolomics.
I second that. Thanks, everyone.
Thanks everybody for joining us today. A special thanks to TMIC and David and Julian and Ian and Rob for your comments today and your answers to the questions. It's a phenomenal learning to have access to you in this type of form. If you do have any other questions for the panelists, please feel free to contact TMIC directly or to send it to BioAlberta, and we will pass those questions along. For further events like this, please watch BioAlberta's event page or our LinkedIn site or the TMIC site as well. We have 2 more panel discussions coming up next week, and I believe the week after. So stay tuned for those. Feel free to register, and we would look forward to seeing you in the near future. Until we see you again, thanks for supporting BioAlberta. Thanks for coming today. Stay safe, and as I always say, trust and believe in science. Thank you very much.